Hi, my name is Gwanghoon Jang, and I'm in Korean University, Department of Computer Science and Engineering. My presentation is Transcriptome phenotype-based drug repurposing framework for discovering COVID-19 therapeutic agents. Let's begin with the global situation of COVID-19. This graph, provided by WHO, shows the number of COVID-19 confirmed cases and deaths worldwide. As of June 30, COVID-19 caused about 10 million confirmed cases and 500,000 deaths. As you can see, the number of confirmed cases is steadily increasing. And furthermore, COVID-19 is highly contagious and causes severe pneumonia. This situation is an unprecedented global health crisis. So that is why the development of COVID-19 therapeutic agent is urgent. To date, COVID-19 drug development has been based on targeting the pathway of the COVID-19, like remdesivir or chloroquine. But this approach has a limitation that it can't be developed if the mechanism of coronavirus is not fully identified. So we propose a novel framework to discover drugs even without fully understanding the mechanism of the virus. The framework discovers drugs that can make return the virus-infected gene transcriptome phenotype to its normal state. This is our model structure in the training process. We exploit the 1,000 dataset provided by Connectivity Map as a training dataset. The 1,000 dataset consists of a drug and the transcriptome perturbation induced by that drug. In the input layer, drug data and the drug-induced transcriptome profile data get embedded to vector. We utilize ECFP for modeling drug chemical structure, which is very famous topological fingerprint for molecular characterization. And we use Z-normalized transcriptome profile vector, which means the relative expression of 978 landmark genes. Embedding layer consists of multilayer perceptrons with drug vector and gene expression vector as input features. Each output vector is mapping into the same embedding space. This is our model in the drug discovery process. First, we make a reverse coronavirus-induced transcriptomic profile. It implies a transition from COVID-19 infected state to the normal state in the transcriptome level. And we Z-normalize the reverse transcriptomic profile vector data with 978 landmark genes as input. In the embedding layer, gene expression data is mapping into the embedding space. Then, we score cosine similarity between reverse coronavirus-induced embedding vector and 6,883 world-approved drug embedding vectors. Then, we can prioritize candidate drugs by cosine similarity, so we can make candidate drug list. This is how our framework works. This is the top 11 candidate drug list of our framework. Among them, 6 drugs are reported to have an association with COVID-19. Lenalidomide ranked at 3rd place, recently entered into a clinical trial phase. And enzalutamide and apalutamide, which is known as androgen receptor, is recently reported to be a promising target of COVID-19. And rucaparib, which is known as PARP inhibitor, is entered into the experimental phase as a potential therapy for coronavirus. So our conclusions are the transcriptome phenotype-based drug repurposing framework can discover drugs without fully understanding the mechanism of the disease. And we suggest 11 candidate drugs as a therapeutic agent for COVID-19. And among them, androgen receptors can be potential inhibitors of COVID-19. If you have any questions about this presentation, please do not hesitate to contact us via this email. Thank you.